So thank you for the introduction. And I'm going to talk on this, really, as Alan said, starting with studying pain in humans, and then drawing back and trying to understand molecular mechanisms of sensitization within the nociceptive system. Now, I'm guessing not everyone on this call is a pain scientist. So actually, I'm going to take a little step back first and just give some introductions to the rationale as to why I started this series of experiments and how we identified this target, sodium-calcium exchanger 3. So I think, as many of you know, we have a specific neural circuit to detect tissue injury, and this unfortunate man is hitting his thumb with a hammer, and that's going to activate specialized neurons called nociceptors that are going to transmit that information to the central nervous system. They will synapse in the spinal cord, and there's complex intraneuronal circuitry within that spinal cord that will modulate that information and transmit it to a distributed cortical network, sufficient activation of which will give rise to the perception of pain. Now, acute pain is important. It's protective and very highly evolutionarily conserved. The real clinical problem is chronic pain. This is a picture, it was one of Frida Kahlo's pictures called The Broken Column. She suffered a spinal injury in teenage years, and then we had really nasty neuropathic pain, which is represented with these needles in the skin for the rest of her life. And chronic pain, unfortunately, it's one of the biggest challenges to human health. It affects about 20% of the population. And one of the things, there are many pathological processes that go on that lead to the development of chronic pain, but certainly sensitization at multiple levels of the nervous system, whether it's the nociceptors, the spinal cord, or brain processing, is really important. So we need to understand and get a handle on those sensitization processes. Now, one way you can test the function of different components of the sensory nervous system is quantitative sensory testing, which is essentially where you're going to be giving stimuli, not always a hammer, but stimuli to the periphery, and correlate that with what people perceive. So that could be a thermal Peltier device, such as a heat pane. It could be a mechanical probe. It could be a pressure probe. And you're going to give defined stimuli and relate that to the perception people report. You can get more complicated, and I'm going to come on to this. So you could, for instance, look at the response to repetitive stimuli, and that's going to be a theme of the talk today. You can look at pathological phenomena. So, for instance, in normal people, brushing the skin with a cotton wool swab or with a brush is not painful, but unfortunately, in some chronic pain conditions, that can actually evoke the perception of pain, which shows very abnormal processing within the sensory nervous system as a result of sensitization. And finally, you could give chemicals, either mustard oil here on the left, or capsaicin, the active component of chili peppers, and you can apply those to the skin, and that will directly activate nociceptors and can also cause sensitization. And the reason I'm going through this detail is to show that there are different aspects and facets of the nociceptive system which you can test with quantitative sensory testing. And another general point I'm going to make, because we are going to come on to human genetics, is there have been a number of twin studies, and this one I particularly like, particularly because it came from my mentor, Steve McMahon, on a twins cohort in the UK. And the reason I like this study is because they studied twins, and they used the conventional comparing association of traits in dizygotic versus monozygotic twins to work out heritability. And what they showed is there is a significant heritability, that can be as high as 0.5, and it does vary between different sensory modalities. So there clearly is, in your basic perception of noxious stimuli, an important genetic component. And as I'm sure many of you know, that our technologies for understanding the genetic basis of really any phenotype has grossly evolved over the years. And really, this is a famous graph showing that very rare variants have very big effects. That would be Mendelian disorders, but most variation in the population is going to be these common variants. These are variants that all pretty much all of us are carrying polymorphisms that are attending this conference, and each of those will have probably quite a small effect size, but can give us great insight into biology. And so actually the kind of the core of this study came from a collaboration I'd had for some time with the University of Antioquia in Medellin. And actually it started because we were studying some very rare pain disorders. But we realized that there is some real characteristics of this population that make it very useful for and helpful for genetic studies. And this is kind of a paper from my collaborator, Andres Ruiz Linares, where he was looking at the genetics underlying a number of phenotypic traits and making the point that in South America, and particularly in Colombia, this is an admixed population, and you have genetic components from European ancestry, native Indian ancestry, and African ancestry. And so you have this huge genetic variety, which makes it actually very powerful for genome-wide association study. And at the same time as I was interested in pain, we realized that they were capturing participants, recruiting them in a study to look at phenotypic traits. So why didn't we actually add pain phenotyping onto that? And I'm not going to go into the full details, but we set up an experimental model where many of those facets that I showed you at the beginning of quantitative sensory testing, we were undertaking in healthy volunteers and participants. And in particular, we weren't just looking at the kind of basic pain thresholds, but we were trying to understand sensitization within the sensory nervous system. We were doing that both by giving repetitive stimuli, and I'll show you that data in a minute, and also by giving a chemical mustard oil, which directly activates nociceptors to try and sensitize the system. And I'm not going to show you all the data from the study because we don't have the bandwidth to do that. But I just want to make the point that this took some validation. Quantitative sensory testing has to be done in a very controlled way. So we had pretty good test and retest data. And we realized with the power calculation that we needed probably between 1,000 and 2,000 participants to start seeing effects on a genome-wide association study. But let's get to the crux of what I want to speak about today. And that's the fact that you will probably intuitively know yourself, but if you were to give a repetitive noxious stimulus, in this case a mechanical probe, and ask people to rate their pain perception with repetitive stimuli, actually, unlike many other sensory percepts such as vision, but with pain, if you give repetitive noxious stimuli, actually pain perception increases quite significantly. And that's temporal summation. Just with a 1 Hz stimulus, you will find that pain perception is higher with the final stimulus compared to the first stimulus. And that's temporal summation, but we can also call it perceptual windup. Intuitively, that makes sense. There's a windup of this sensory percept. And Louis, who is a dentist in Colombia, did the heroic job of recruiting, well, actually, he's just under 2,000 participants now with this experimental pain model. And we've, if I show you the data, which is what this wind-up looks like. So this is a wind-up ratio, and essentially it's a ratio of the kind of pain perception at the end of the train of stimuli versus the beginning. So anything over 1 indicates wind up, and you can see that most people do wind up, and that there's great variation. Some people show very little wind up, but some people show a large degree of wind up. So like any phenotypic trait in the population, there is variation, and that means that we can then try and understand the genetic basis of that. And so really this is trying to get a handle on what's controlling sensitization following repetitive stimuli to the nervous system. And when we did that, we did a genome-wide association study, and this is the kind of how you would present that data. This is a Manhattan plot, essentially showing across the whole genome, single nucleotide polymorphisms, and the degree of significance of relating those to a particular trait. In this case, it's the wind-up ratio. And we saw 2 signals. We saw this S100A16, which I'm not going to talk about more here, but it's calcium binding protein. And then this highly significant signal in the locus is SLC8A3, and that encodes for sodium calcium exchanger 3. This red line indicates that it meets genome-wide association significance. The good news was we could then replicate that in a separate cohort, which we recruited in Colombia, and it did replicate. And actually, we also replicated at gene level in a cohort where Luda Diachenko had collected wind-up data in a completely separate cohort in North America. So we had an interesting genetic signal which replicated across multiple cohorts, which related to wind-up, perceptual wind-up in humans. And kind of just to show you that in a bit more detail, so this is what's called a zoom plot. It's showing you where the single nucleotide polymorphisms are. So these are not coding variants. These are variants within introns within the gene that are near an enhancer region, and the most likely mechanism by which they're altering functional properties is through changes in expression of this gene. What's interesting about this particular single nucleotide polymorphism that we identified is it's only present in Africans. So had we done this genome-wide association study in a Caucasian population, we would have completely missed this association. That's a growing theme in human genetics. The more of the minor alleles you have, the higher the wind-up ratio. So there's a dose-related effect. And as I've said, this gene encodes for sodium calcium exchanger type 3. And what this does is this extrudes, particularly with high levels of calcium, it extrudes calcium for the cell. So calcium is exported and sodium is imported. And of course, we know that calcium is important for many neuronal functions. So this was intuitively and immediately interesting to us. So just kind of to come back a bit to the basic neurobiology. So and now I'm really going to draw focus to the spinal cord. And that's because there was pre-existing data that the spinal cord is a really important locus for this sensitization and wind-up phenomena. And we knew that because Lorne Mendel and Pat Wall had shown with recordings in the cat, actually, that if you give the same electrical stimulus activating nociceptors in the periphery and you repeat it, that the response of dorsal horn neurons within the deep laminae of the spinal cord progressively increases. And this is a raster plot, and essentially each of these is an action potential. But rather than try and show this rather complicated raster plot, you can hear this. So the stimulus is the same, recording extracellularly from dorsal horn neurons. And you will see as the train of stimuli increases, that response will... I think you can agree that's wind-up happening in real time. You could see as that train of stimuli went on, each individual stimulus to the periphery was the same, but you're getting a progressive increase in the response of the dorsal horn neurons to that input. And that is spinal cord wind-up at an electrophysiological level, and you can see why we think that might very nicely link to perceptual wind-up in humans. So now we could start trying to explore that using animal models. Just to say that there has been a lot of work on the synaptic basis of wind-up, and essentially that relates to an interaction between neuropeptides being released by primary afferent nociceptors and glutamatergic transmission, and the activation ultimately of the NMDA receptor and ingression of calcium is thought to be very important in this process. And this is just data from one of Clifford Wolf's papers showing, using patch clamp analysis, again, this progressive depolarization and then increased response to repetitive stimuli. And again, a lot of work has gone into the idea of increasing calcium ingress into dorsal horn neurons within the spinal cord, but very little work in what then happens to the handling of calcium within those neurons. So again, sodium calcium exchanger 3 was a very interesting and important target for us to explore. So first thing to know was sodium calcium exchanger 3, is it expressed within the spinal cord? So we can check that in mouse tissue, and that's what we did. Essentially, whether we use markers for excitatory interneurons, VGLUT2 here, or GABAergic interneurons, or glycinergic interneurons using reporter mice, and we performed in situ hybridization for NCX3, we can see it's expressed in all of these populations. So it's expressed with interneurons within the dorsal horn of the spinal cord. It is also expressed within projection neurons, which we've marked here with a marker FOX2A. to kind of summarize kind of a lot of data and analysis, essentially, NCX3 is broadly expressed within the dorsal horn of the spinal cord. There is some increase of expression as you go into the deeper laminae of the dorsal horn of the spinal cord, and that kind of fits with the fact that actually windup is particularly marked within the deeper laminae. And we also found that it was expressed both within interneurons and projection neurons. So it is expressed within the right locus to mediate these effects. And we can then take luckily, Andrei Hirschulz and Sophie Sokolov had already made mice which don't express NCX3. So we could then try and validate these human genetic results in the mouse. And when we study pain behavior in mice, we can look at evoked responses to noxious stimuli, whether that's a pinprick or a von Frey probe, or whether it's a thermal stimulus in the form of Hargreaves test for response to thermal stimuli. And this did not change. And that is not a surprise, because in the human genetics, there was only a relationship between the NCX3 locus and temporal summation or windup. There was no relationship to pain thresholds in response to noxious thermal or mechanical stimuli. But the other thing we could then do in mice is to look at more markers of tonic pain-related behavior. So one of those is the formalin test, where formula is injected into the paw, and the not offensive behavior, the licking and wiping of the paw by mice, can be assessed. And there's two phases of that. There's an acute phase, which is thought to be just the acute response of activation of nociceptors. And then there's a second phase, which is thought to be largely or partially spinally mediated sensitization within the spinal cord. And we could see in mice with either heterozygote or homozygote mutant alleles for NCX3 increased response in the second phase of the formalin test, and that's just quantified here. And so that does fit with increased sensitization at a spinal level. We could then also look at a model of nerve injury, which we call chronic constriction injury, which is a cuff or a number of sutures placed around the nerve, and then look at the response to mechanical stimuli, which is sensitized in wild-type mice, but even more sensitized in those mice lacking NCX3. So what we're finding is a lack of NCX3 is associated with enhanced pain-related behavior in more tonic models of pain in the mouse. And we could then look at calcium handling within, firstly, we looked at cultured dorsal horn neurons. And we can assess using fluoromediated calcium imaging and see that in the mice lacking NCX3, both the baseline calcium is raised, the peak response to depolarization with potassium is enhanced, and the rate of the kind of decay constant, which is going to be a measure of calcium extrusion, is slowed. So all of these aspects of calcium handling seem to be altered. Now, this is looking in culture, and this does give us the advantages. We can get a closer handle on some of these more specific features of calcium handling, but obviously it'd be better to look in vivo. And we were able to do that with Steve McMahon and Kim Chisholm, because they developed a protocol where they could label projection neurons from the spinal cord using so that they would express calcium genetically encoded calcium indicator. And then we could essentially do the same experiment that we've done many times already, which is to stimulate with electrical stimulation nociceptive afferents and look at calcium handling within these projection neurons. And the result was quite stark, so that there was some increase in baseline calcium, which again would fit with the fact that we have a deficit in extrusion of calcium, but a really enhanced response to stimulation. And you can see these much bigger peaks at whatever stimulus, whether you're looking at a low or a high frequency of stimulation, much higher calcium within these dorsal horn neurons. That's calcium imaging. And obviously, when I was playing you that recording, we were actually looking at action potential. So what happens if we come back and look at action potential? So this was work by Ryan Patel and Tony Dickinson. And this is the paradigm that we were discussing before, which is recording from dorsal horn neurons, recording their action potentials in response to stimulation of electrical stimulation of nociceptors. And actually, one of the interesting features here is that they've been looking at a frequency of 0.2 hertz, where there is some wind up, but it's pretty mild in the wild type. Whilst in animals which have one mutant locus for NCX3 or they're homozygous for mutant NCX3, we can see this much more marked wind-up. So this is comparing the 16th stimulus to the first. You can see there's a mild increase in the wild type, but massive increases in the heterozygous and homozygotes. And it was very clear that you had this very marked after discharge as well. So we not only have we seen a change in calcium handling, but clearly that has an impact on the excitability of these dorsal horn neurons as well in vivo. Just to say that that's recording from the dorsal horn. If this man is hitting his hand with a hammer, that will naturally evoke a flexion reflex. We withdraw from noxious stimuli, and that can be quantitated by recording from the efferent part of that pathway, which is to record from motor neurons. That's the flexion reflex. And so we can, again, look at that electrophysiologically, and the story is the same, that whether we look at a low frequency, middling, or a high frequency, we're seeing much more activation of those motor neurons in response to the same input. So the flexion reflex is enhanced across a range of frequencies, and as it were, so the output of the spinal cord in terms of the flexion reflex is also sensitized with these animals that don't lack functional sodium-calcium exchanger type 3. What's the kind of flip side of this? So I hope I've convinced you that we've found a genetic target in humans. We showed in mice that don't express NCX3, enhanced pain-related behavior and increased excitability and wind-up within the somatosensory nervous system at the level of the spinal cord. Can we then do the reverse and overexpress NCX3? So the idea here is we've taken the neuronal isoform of NCX3, we've put it into an adeno-associated virus. If we take NCX3 and we can see it, this is just a kind of proof of concept. If we express it in hex cells and we can see it where it should be, which is on the surface of those cells, we can find that it speeds up calcium clearance if we just load these cells with calcium with ionomycin. So the next step was to say, okay, let's now enhance activity of NCX3 within the nervous system. And to do that, we worked with Andrew Todd and his excellent team in Glasgow, where we could inject this virus into the dorsal horn of the spinal cord, and then we could see what impact that has on pain behavior. Our hypothesis here is that can that reduce sensitization at a spinal level because we're enhancing calcium extrusion. We can see nice expression of NCX3, which was tagged with a MIC tag selectively within the dorsal horn of the spinal cord. It doesn't spread to any other parts of the nervous system, so we can really localize this to the dorsal horn of the spinal cord. And as expected, acute pain thresholds did not change, and that's because, as I've tried to elucidate, this is really a gene that's having an impact on sensitization at the level of the dorsal horn of the spinal cord. But when we look at the Formelin test, where this second phase of the Formelin test is pain-related behavior really relates to sensitization within the spinal cord, we could reduce that pain-related behavior in that second phase significantly. Really pointed to the fact that we can modulate this system in either direction, and if we can increase activity of sodium-calcium exchange, we can reduce tonic pain-related behavior. And I think that's kind of does give some interesting prospects for whether you could subsequently modulate this therapeutically. I don't think gene therapy necessarily is going to be coming as a treatment in humans soon, but you could, for instance, think about small molecules that could enhance activity of NCX3. One challenge of doing that would be that there are a number of NCXs, NCX1 and NCX2, and people have this has been a therapeutic target, for instance, in stroke in other contexts, and the challenge will be to get selectivity just for NCX3. Other kind of future directions are that actually, I've shown you a fraction of our data. So we, as I tried to point out, we were looking at a number of different sensory modalities within those participants within Columbia, and we're finding other genes which are related to other aspects of sensory function. So I think this is going to be quite a rich vein of starting with the human and then coming back and validating in mouse models. Those were healthy participants. The other thing we're looking at now is trying to get more detailed information of sensory function in patients. And I don't have time to go into details, but for instance, we can take patients with neuropathic pain and we find that we can get complex patterns of sensory dysfunction. There are some people that have predominant sensory loss. There are others that have mechanical hypersensitivity. There are others that have thermal hypersensitivity. And we're now trying to link particular genetic variants with these particular patterns of sensory function, for instance, to see if not only can that help us understand pain in patients, but also can that help us maybe target our therapies more effectively than a personalized medicine approach. So I'm going to end there, and I hope I've convinced you that GWAS in humans has provided an unbiased means to study novel gene variants and understand sensitization within nociceptive circuits. One take-home message is I think the genetic architecture of different modalities will be distinct. I don't think there's going to be one single pain gene that's going to affect all modalities. I think different genes are going to have impact on different sensory modalities. And today I've honed in on the sodium-calcium exchanger, which I think is a key determinant, not only of perceptual, but also of electrophysiological wind-up in the human. And I think it may be amenable to therapeutic target to reduce sensitization of these nociceptive circuits in humans. And I will end there. I'll thank my really wonderful lab that have done so much of this work. I'd particularly like to highlight Teodora Trendafilova, who was a DPhil, a PhD student with me, and she really drove much of this work and did a wonderful job validating all of this human genetic studies in the mouse. And particularly, I'd like to thank Kaustu and Andres Ruiz-Linares, who were collaborators on the human genetics. I'd like to finally end by thanking my funders, and thank you for your attention.